Hello. In last lecture, lecture one, we have talked about the classification. Biological classification, we done with our five kingdom classification and six kingdom classification, which is a accepted five kingdom classification. Now we are moving to the kingdom Monera. Yes, the first classification in the five kingdom. We are talking about the Monera in detail. So let's start in that. The great enthusiasm. So here we go. So as I told you, we are going to talk about the kingdom Monera, the first one. So what are the characteristics? So before we go in, what are the characteristics of that? So the first is their prokaryotes include all the bacteria. Please make sure you're reading this. All the bacteria are exclusively found in Monera. Then they are ubiquitous. Okay. Then they are double-stranded circular DNA. Now let's talk about this too in very detailed manner. Here we go. So when you talk about the ubiquitous, they are what? They are found everywhere. Found everywhere. Yes, they are found everywhere. Now I'm going to change the color over here. I'm going to take a yellow. It will be the very helpful, right? Yes. Then bright color. Okay. So found everywhere, it is ubiquitous. Then that double-stranded circular DNA. Circular DNA like this. Yes. Why they have a circular DNA? Because they are not that much well developed. Exactly correct. Then it's a double stranded, one strand, second strand, circular one, right? Yeah. Then we have a membrane bound cell organelle absent. Membrane bound cell organelle is absent. Why? Again, bacteria they well developed and they are prokaryotic. That is the reason. Yes. Then we have a rigid cell wall. Yes, they need a protection for that. Okay. For the environmental protection, a rigid cell. They have a 70S ribosome, 70S. Okay. They may be motile. If they are motile, they have a flagella. Like suppose this is a cell and you want to move. So they have a flagella. It's called what? Flagella. Okay. Okay. Very good. If they are non-motile, then obviously they do not have a flagella. Correct. Very good. Then they have a nutrition mode is heterotrophic mode of nutrition. That is the saprophytic or parasitic or sometimes autotrophic as well. So let's understand this one diagram. That is the first animal cell is there, right? And this viruses, and this is the bacteria. Are you getting my point? So this is the bacterium we are going to talk about because exclusively bacteria found or fall under the monica. Okay. So let's talk about this diagram. Okay. So when you talk about they have a what I'm talking about, they have a cell wall, which is rigid one. Exactly. So rigid cell wall is there, right? So first it's found as a capsule. So they are found under the capsule-like structure, not exactly the square one, like this capsular structure. Okay. And inside the other things are found. Okay. So here we go. Okay. So we have understood that very well, right? Let's talk about the things. So they have a cytoplasm. This is the granular structure cytoplasm. They have nucleoid. See, DNA, it's called nucleoid. Yes, nucleoid. Please remember. Then they have a plasma membrane as well, right? And they have a ribosome. Which one? 70. Very good. They have a cell wall, which is rigid one. Okay. Then capsule is there. Then pillars. What is this pillars? What is this pillars? See, so we are aware about the cilia. In paramecium, we have a cilia for locomotion, right? Small hair-like structure. Here, they have a similar kind of thing, but they are called pillars. Single is pillars or otherwise cilia. Then they have a flagellum for a direction, for a locomotion, for a locomotion, smooth locomotion. Pilius for the absorption and for what? Conduction. Okay, so this point is very much clear. Now we are moving to the next one. So we have done about the bacterial structure. Yes, we are still talking about the kingdom Monera. Yes, we are talking about the kingdom Monera only. So classification of the bacteria all found in a Monera. So based on their shapes, we have a cocci. Cocci means what? Can you see that? A circular structure, a round structure. That's called cocci. Then we have a bacillus. Bacillus is what? This is a spore. It is found over there, and this is the bacillus. You can draw bacillus like this. And we have a flagellum found in a spirilla. Spirilla means this kind of structure, spiral-like structure. Okay, and they have flagellum also from both the sides. Okay, for the motion. Then we have a comma-like structure. Can you say it's resemble like comma? Yeah, that is a vibrio. Example is vibrio cholerae. Yes, that is causes the cholera. Exactly correct. So that is the vibrio. So this is on the based on the shapes. Okay, bacilli, spirilla, and the vibrio. This point is very much clear. Yes. Can we move to the next one? Yes. Here we go. Now, kingdom Monera. When you talk about the kingdom Monera, the first point has to be in your mind is bacteria. Yes, it's a bacteria. So reproduction in a bacteria, there are two modes, asexual and sexual. When you talk about the asexual, first found is a binary fission. Binary fission, the moment what you are recalling, one cell undergo the two different daughter cells. Exactly correct. So the process in which the parent cell divides into two independent daughter cells. Why it's called daughter cell? It should be called son cell also, right? No. Why daughter? Because they're identical to each other. That's why they use the word daughters. Very correct. So can you see that this cell is undergo the binary fission, cleavage at the center, and they is forming the two new daughter cells. Then we have a sporulation. So what is sporulation? Here we go. The process in which the parent cell produces the spore and the spores. When you go for the class 12, there is a pan cosmic theory. That is, we are like aliens, right? So we're going to study about that. So there is also the word spore is there. What is actually the spores? Which giving the viable life, new life. That is a spore. Okay. That is carry the tiny pH. That is a spore. So spore, each of which can develop into the independent organism. That is called sporulation. The process in which the parent cell produces the spore or spores, and that spore, whenever it gets a viable condition, suitable condition environment, it is grow into the independent organism. Like this is a cell produces spore. Okay, this is the endospore, that is inside the cell. Okay, present within the cell. If there is an exospore, it is outside the cell. Okay, that cell detaches, or that spore will be detaches from the parent cell whenever they found the viable condition. They do this thing. But why they process the spore? Why they perform the spore only? Why? Because spores. Okay. So I will tell you one thing. There are the concept of dormant cell. You are aware about it, right? What is dormant cell? For a particular period of time, they act like a dormant. Means after sometimes they get a viable. Means what? Why plant produces the dormant seed? Because he has to protect those seed. He want to pass on that whatever the wrong climatic condition or the unfavorable conditions are there, that plant want their seed to pass from that thing, and then it give rise to the another plant for a protection purpose only. Yes or no? Yes or no? Similarly, whenever there is a unfavorable conditions are found, the bacteria produces a spore for maintaining the generation. Correct? Exactly. Then, when we have a sexual reproduction, it's called conjugation. So, what is conjugation? We know that conjugation base and pair in chemistry. So, they are related only, right? Conjugation is related, but in biology, it refers to the process by which one bacterium transfers its genetic material to the another bacterium. So, already cells are present side by side, and they are transferring the genetic information. And so how they can transfer the genetic information here is not written. So they are called as sex pili. Okay, they are called as sex pili. These are the things, right? Yeah. So via that they transfer the genetic. That is called a conjugation. So binary fission, sporulation, and conjugation. These points are very much clear to you. Yes. Now we are moving to the forward. Again, Kingdom Monera, yes, we have to study that in detail, right? So we have to study it properly. So we have a Archaebacteria, then we have a halo. See, in Archaebacteria, we have a halophiles. Say halophiles? No, that is this is not a halo. This is halo means what? Yes, salty conditions. Salty conditions, yes. Then thermoacidophiles, where temperature and acids are found. These are the bacteria, they can live over there. And the methanogens, in a methane gas, they can produce these archaebacteria mostly found in a harsh condition. Harsh condition. Great, no? They are found in a harsh condition such as halophiles, thermoacidophiles, methanogen. So right now, I'm just doing the category. We are going to study in detail with the example as well. Yes. Then eubacteria is also called a true bacteria. So whatever study are the false bacteria, no. This is the true bacteria because they have the identical characteristics as a bacteria. It consists of cyanobacteria, nitrogen fixation. Yes. Then we have a heterotrophic bacteria. Then we have a actinomycetes. We have a mycoplasma. So let's study in detail. First come Archaebacteria. The moment I say Archaebacteria, what is the first point come in your mind? The thing is, they are a harsh condition bacteria. They are the most primitive group that occurs in the extreme habitats. The name itself suggests their cell wall is made up of the pseudo peptidoglycans. Please underline this or make sure when you are hearing this, make sure you're writing somewhere. That is that is pseudomurine. Pseudomurine or also called as pseudopeptidoglycan. That's why they are very much readily available or they can survive in the extreme habitat because of the pseudopeptoglycan, also called as the pseudomurine. So the halophilids, first, they are found in a saline environment. They have been observed in the Great Salt Lake. USA. Even there is salty lake, no organisms will survive such an extreme condition, but still they are fine. So they are called halobacterium. Then we have a thermoacidophile. They are found in environments having high temperature and the low pH. High temperature, almost all bacteria will go to the death, right? Yes or no? Almost they are dead. But high temperature and low pH, such a environmental, drastic environment they are surviving. They have been observed in a Yellowstone acid pool in USA. They are called sulfolobus. Sulfolobus. Okay, then we have methanogens. The name itself suggests methane is there. Okay, so they are found in a gut of ruminants. The gut of ruminants? How come? Because they are the one actually digest the things, right? Over there. So they're responsible for the production of methane from the dung. So that's methanogens. They actually kind of give out the methane. So methanobacteria. So these are the archaebacteria. Halophiles, which is saline environment found. Thermoacidophiles, which is found in a high temperature, low pH, that is sulfolobus. And methanogens found in the gut of ruminants. What is ruminants? The cows, buffaloes. Okay, that's called the ruminants. They are responsible for the production of methane from the dung. That's called methanobacteria. This point is very much clear, right? Am I going slow? No, I'm not going slow. Actually, I want to emphasize on those topics. That's why I'm just talking about it. Okay, so let's go for it. Now, kingdom Monera, eubacteria. They're also known as the true bacteria. Yes, they're true bacteria. Their cell wall is made up of the peptidoglycans. They can be of following types depending on the mode of nutrition. So, what they are? Cyanobacteria. We are talking about the cyanobacteria. So there are true bacteria like chemosynthetic autotrophs, heterotrophic, actinomycete, and mycoplasma. Right now, I'm talking about the cyanobacteria. So also known as the blue-green algae, nitrogen fixation. Yes or no? Yes. They are photosynthetic autotrophs that contain chlorophyll A. Which one? Chlorophyll A. Please pay attention. Chlorophyll A. Then phycocyanin, second constituent, and the phycoerythrin, red color, right? Okay. So blue and green algae, why they are talk about? Because they have the pigment phycocyanin, that is of blue color, and the erythrin is for reddish color. Okay. So showing the gliding and the oscillatory movements are covered by the gelatinous sheet. So they have a gelatinous sheet around them. Okay. Like these are the like this. Okay. Okay. Then found in the freshwater, marine, and the terrestrial regions. Examples: Anabina, nostoc, both are the heterocyst as well. Once again, what is the heterocyst? Now I'm drawing this diagram with for you. So all cells are similar, but one cell found this kind of structure and which appear different from the other cell. This is called as a heterocyst. That means hetero means not homogeneous, I mean different. So this only one cell is responsible, or that is called as a heterocyst. Okay, so it's found in an anemone. Three types are found, like spirulina, microcystis, and nostoc. So what is that? Let's see. Okay, so spirulina, it's a unicellular. Microcystis, colonial, and nostoc filamentous. So unicellular means they are found like this. Microsis colonial matlab they found in a colony, and filamentous it's formed in a filaments like that. Okay, so this is all about the cyanobacteria. Now, eubacteria. The true bacteria is what? True bacteria, right? Yes. So, chemosynthetic autotrophs, this bacteria oxidize inorganic substances such as nitrates, nitrites, and yeah. Energy thus release is used for the ATP production. Heterotrophic bacteria, most abundant in nature. Heterotrophic are most abundant. Most of them are saprophytes. Yes, that's the truth. So this can come for the statement. A certain reason. Most of them are saprophytes as a heterotrophic bacteria. They're useful in a curd formation from the milk, nitrogen fixation from the root of leguminous plants, and production of the antibiotics. Some are the pathogenic, some are dangerous, some are useful. They causes the disease in the plants, animals, and humans like cholera, typhoid. These are the heterotrophic bacteria. Okay, this is true bacteria, eubacteria. Now there are some points that is remaining: actinomycetes and mycoplasma. So actinomycetes, there is mycelial bacteria that helps in a decompose the organic material. That is the chitin. Chitin is made of cell wall, right? The protein which is responsible for the cell wall formation. That these are the one who actually die, disintegrate, destroyed, right? Those are the actinomycetes. Okay, they are mycelial bacteria. Then they produce the antibiotics. So actinomycetes, antibiotic. Actinomycetes, antibiotics. Remember this. They commonly found in a soil and aquatic region, both freshwater and marine. Example is streptomyces. Example is streptomyces. Now I'm talking about the mycoplasma. Mycoplasma is called Joker-like structure. Why? Let's start understand. First is the smallest living cell, very small living, also called a PPLO, that is a pleuropneumonia-like organisms. Lack of the cell wall and are non-motile. So they don't have a cell wall. They are non-motile. They are very small. Okay, so they exhibit the pleomorphism and can survive without oxygen. So it is a small, it is a PPLO like pleuropneumonia like organism, lack of the cell wall, they are non-motile, they don't need oxygen, and they exhibit a pleomorphism. So this is called the jokers. Yes, yes, yes. And they pathogenic to the plants and the animals. Because of this nature, they are pathogenic. Okay, so that is the kingdom Monera eubacteria. In that, we talk about the actinomycetes and the mycoplasma. So here we concluded eubacteria, we cover cyanobacteria. Yes, we cover the chemosynthetic autotrophs. Yes, heterotrophic bacteria. Yes, actinomycin and mycoplasma. So here we cover the kingdom Monera in detail. Okay. So let's talk about kingdom Monera. Archaebacteria, harsh condition bacteria, halophiles, salty lakes, thermoacidophiles, low temperature, no. High temperature, low pH. And methanogens found in the ruminants of the gut ruminants, right? And eubacteria, cyno, heterotrophic, actinomycete, and mycoplasma. Cyanobacteria, lubrine algae, heterotropics. We are already aware about actinomyces. They are called antibiotics, and mycoplasma. They are very small in size. Correct? We understood that, right? First, they're very small in size. Then second one, they are PPLO, right? Yes. And they are live without oxygen. No oxygen required. Okay. And they are for dangerous for the plants and animals because of their size. Okay. So we have covered the kingdom Monera in very detailed manner over here. So I hope you understood this very well. Okay. Thank you for paying attention.